Not for video. Welcome back to my new amazing video. My videos are fact-based, made for curious minds and knowledge lovers. If you spot any mistakes, please comment. It helps me improve and keeps the facts accurate. Some may love cinematic visuals, but my focus is purely educational and informative. And don't forget to subscribe my channel, like video and comment. Let's start the video. What if the very thing designed to create life, a mother's own blood, turned against her unborn child? For 40 years, a medical ghost haunted maternity wards. It was an invisible enemy that could turn a pregnancy into a tragedy and a life-saving transfusion into a fatal mistake. Doctors knew something was there. They saw the devastation it caused. But they couldn't find it, they couldn't name it, and they had no idea how to stop it. It was an impossible blood type that seemed to break all the rules. This is the story of how cutting-edge science finally solved that 40-year-old cold case, cornering a phantom hiding in our very veins. The 40-year cold case. Our story starts not with one patient, but with a puzzle that took decades to solve. It all began back in 1982. In a lab, a scientist is running a routine cross-match, mixing a patient's blood with a donor's. But the result is anything but routine. The patient's blood violently attacks the donor cells. This wasn't a simple ABO mismatch, this was something totally different. The patient's immune system had built a mysterious antibody, a tiny protein missile programmed to destroy what it thought was a foreign invader. Scientists named the target of this antibody era. They found it was a high-incidence antigen, which is just a fancy way of saying nearly everyone on the planet has it. So, for the rare person without ERA, their immune system sees almost everyone else's blood as a threat. Finding a compatible blood donor for them would be like searching for a needle in a global haystack. And for years, that's where the story stalled. ERA was just a label for a reaction, a name for a ghost. No one knew what it was actually made of. A few years later, in 1988, another related marker was found, called ERB. Then a third, ER3. They were all clues in a case that was getting colder every year, filed away in a kind of miscellaneous folder because they didn't fit into any of the known blood groups. Think of our blood types as complex ID codes. Most of us know the big two, ABO and the rhesus factor, which is the plus or minus sign next to your type. An Austrian scientist named Karl Landsteiner figured out the ABO system back in 1901, and it was a game changer, finally explaining why early blood transfusions were such a deadly gamble. But those two are just the opening chapters in a massive encyclopedia of human blood. The International Society of Blood Transfusion, or ISBT, currently recognizes 48 official blood group systems. Each one is defined by genes that create the antigens, the little protein and sugar, flags, that decorate our red blood cells. These flags are how our immune system tells friend from foe. For 40 years, the antigens were flags without a country, orphans of the blood world, known only for the destruction they could cause. And the true cost of this mystery wasn't measured in the lab, but in the devastating silence of a hospital room. The stakes of war in the womb, while a mismatch during a transfusion was a serious risk. The real front line of this medical mystery was pregnancy. The worst reactions were seen in cases of something called hemolytic disease of the fetus and newborn, or HDFN. HDFN is a cruel condition where a mother's immune system accidentally tags her own baby's blood cells as foreign invaders and launches an attack. This happens when the mother lacks an antigen that her baby has inherited from the father. Her body then creates antibodies that are small enough to cross the placenta. Once they're in the baby's circulation, these antibodies lock onto its red blood cells, marking them for destruction. As the baby's red blood cells are destroyed, the fetus becomes dangerously anemic. Its tiny body tries to keep up by pumping out new, immature red blood cells, but it's a losing battle. The baby's liver and spleen swell as they work overtime, and eventually, the heart begins to fail. This can lead to a catastrophic condition called hydrops fatalis, where severe swelling overtakes the baby's body, often resulting in stillbirth. In the case of the antigens, the consequences were tragically real. As part of their investigation, scientists looked at the histories of several patients, including 2 mothers who had powerful antibodies against 2 previously unknown versions of the antigen. Both women tragically lost their babies to severe hemolytic disease. Can you imagine the agony and the confusion? A mother's body, designed to nurture her child, was harming it for a reason nobody could explain. Doctors could see the anemia and detect the antibodies, but they couldn't tell her why. They couldn't find the genetic root of the problem. For these families, and for the scientists on the case, the stakes couldn't have been higher. Finding the genetic basis for this phantom blood type wasn't just about scientific curiosity, it was a race to give families answers and prevent this from ever happening again. The breakthrough cornering the ghost for decades, the tools just weren't there to solve the puzzle. The effects were obvious, but the cause was a complete mystery. Then came the 2010s, and with them, a new era of technology that would finally give science the upper hand. The breakthrough came from a dedicated team at the UK's National Health Service Blood and Transplant, NHSBT, and the University of Bristol. They were armed with something scientists from the 80s could only dream of: advanced DNA sequencing and the gene editing tool CRISPR-Cas9. The team gathered precious, historical blood samples from just 13 people known to have made these mystery antibodies over the last 40 years. This biological library was the key. They used a technique called whole exome sequencing, which let them rapidly scan all the gene coding regions of a person's DNA. Instead of looking for a needle in a haystack, they could now use a giant magnet. They were hunting for a common thread, a specific genetic typo shared by these 13 individuals that could explain why their bodies were making these antibodies. It was painstaking work, sifting through the billions of letters in our genetic code, looking for one critical misspelling. And then, they found it. The eureka moment. Every single patient with the antibodies had a mutation in the exact same gene, a gene called PIEZ01. This was it. This was the genetic home of the antigens. The ghost finally had an address. To be absolutely sure, the team used some brilliant modern science. In a lab-grown cell line, they used gene editing to snip out the PIEZ01 gene. Just as they suspected, the antigens vanished. Then, they put the gene back, and the antigens reappeared. It was definitive proof: the PIEZ01 protein was the carrier. In the process, the team didn't just solve the old mystery, they discovered 2 brand new antigens, which they named ER4 and ER5. It was these 2 that were involved in the tragic cases of hemolytic disease. After 40 years of eluding science, the phantom was finally unmasked, and the International Society of Blood Transfusion officially recognized it as the 44th human blood group system. This discovery is a huge moment in transfusion medicine, a victory won through decades of persistence and incredible new technology. It's a powerful reminder that our own DNA is still full of secrets. If you find these kinds of scientific detective stories as fascinating as I do me a favor and hit that subscribe button and turn on notifications. We explore amazing stories like this every week, and you won't want to miss what we uncover next. The science explained the body's touch sensor. So what exactly is this PIEZ01 gene, and why does one tiny change in it create a whole new blood type? This part of the story is truly fascinating. PIEZ01 isn't just some random piece of code, it's a master mechanosensor. The simplest way to think of it is as a highly sensitive, touch sensor, on the surface of our cells. Its job is to feel physical force, when a cell gets squeezed, stretched, or pushed. This is critical all over your body, but it's especially important for your red blood cells. A red blood cell has a tough life. It's constantly squeezing through capillaries that are narrower than the cell itself. The piezo-1 protein acts like a tiny pressure release valve. It detects the squeeze and lets a few ions into the cell, which helps it regulate its volume so it can deform and bounce back without breaking. It's an incredible piece of biological engineering. In fact, the discovery of piezo proteins was so important it earned the 2021 Nobel Prize in Physiology or Medicine. The antigens, the very things that caused all the trouble, are actually located on the outside of this piezo-1 protein. For almost everyone, the piezo-1 gene builds this protein in a standard, wild-type, configuration. This is the version our immune systems recognize as us. But patients in the study had a rare genetic typo, a so-called missense mutation. This tiny error caused their bodies to build a slightly different piezo-1 protein. To their immune systems, this altered version is perfectly normal. The conflict happens when these two worlds collide. If a person with the mutation gets a blood transfusion from someone with the standard piezo-1 protein, or if a mother with the mutation is pregnant with a baby who has the standard version, her immune system sees that common protein as a foreign threat. It screams, "Invader," and creates powerful antibodies to destroy any cell carrying it. And that's the solution to the 40-year mystery. The blood group isn't about having a strange new protein. It's about having a slightly altered version of a protein that's almost universal. The impossible blood type was really just a rare variant of a fundamental protein, present in only a few hundred copies on each red blood cell. It just goes to show that even the tiniest molecular differences can trigger a life-threatening response. The broader impact from phantom to fact making the 44th official blood group system was more than just updating a textbook. It turned a decades-long mystery into a solvable medical problem, with real-world consequences for patients everywhere. First and foremost, it offers answers and hope to families who have suffered unexplained losses. As Nicole Thornton, one of the lead scientists, said, the most important thing for them was providing answers to the two mothers who lost their babies. For them, this discovery wasn't just data, it was closure. Now that the genetic basis of is known, labs across the globe can develop simple and fast genetic tests. For the first time, doctors can screen expectant mothers for these rare piezo-1 mutations. If a mother has a rare type, her pregnancy can be watched much more closely. Doctors can be ready with life-saving interventions, like in utero blood transfusions for the baby. What was once an invisible threat can now be identified and managed. This also completely changes the game for patients with these rare blood types who need a transfusion. Before, finding compatible blood was a desperate and often failed search. Now, blood banks can screen donors and build a registry of those who are negative, ready to be called upon when needed. It's another step into the era of personalized medicine, where a patient's own genetic code can be used to give them the safest care possible. And the story doesn't end here. The solution to the mystery is a powerful roadmap. There are still dozens of known blood antigens that haven't been sorted into a specific blood group system. The success of the piezo-1 investigation shows that with the power of modern genomics, no medical mystery is truly unsolvable. For 40 years, the blood type was a ghost story told in labs and whispered in maternity wards. It was a puzzle with missing pieces, a medical thriller with a real human cost. The story that started with a strange reaction in a Petri dish in 1982 has now been solved, all thanks to technology that was once pure science fiction. The journey of the blood group is a testament to scientific persistence. It's a story that moves from the heartbreak of loss to the hope of prevention, from a phantom in the blood to a fact in a gene. This discovery not only brought the 44th blood group into the light but also gave doctors the tools to save the lives of the unborn and provide the safest possible care for people with some of the rarest blood on Earth. It just proves that sometimes, the biggest secrets aren't hidden in faraway galaxies, but are written in a single, silent letter of the code that makes us who we are. The ghost has been caught. And for families in the future, that changes everything.